Let's bring on Will Williams, President, CEO, BriaCell Therapeutics Corporation. Welcome to the show, Will.
Thanks so much. Thanks, Michael.
My pleasure. One of the things that I didn't mention is that you're also publicly traded in two different exchanges, right?
That's absolutely correct. In the Toronto Exchange and also the OTCQB.
What are the symbols?
BCT and BCTXF.
All right, great. All right, you go by Will, Bill, William?
Any of those would work. Bill is usually what people call me.
All right, Bill, you're also a medical doctor. Give us some background on you, your accomplishments, and let's go right into what BriaCell Therapeutics does.
Sure. So I'm actually trained as a rheumatologist and immunologist, was at University of Pennsylvania for a number of years, did molecular immunology, translational research, brought some novel therapies into the clinic, and then went over to GlaxoSmithKline to learn how to. And so now had been at GlaxoSmithKline for 7 or 8 years when I switched over to Incyte Corporation. And Incyte, I was head of exploratory development, where I helped introduce molecules like Jakafi into the clinic and was able to be with that all the way through to post-approval and see patients derive tremendous benefit. And then about a year ago, Martin Schmieg, one of the boards of directors members of BriaCell, introduced me to the company, and I just saw a tremendous potential in what BriaCell has to do great, provide great benefit for patients, especially with breast cancer, but also other cancer types.
All right, now and thank you for that. Let's dive into the history of BriaCell's targeting immunotherapy platform and what is the clinical, what is it saying in the clinical data to you right now?
Right. So BriaCell has a very interesting history. The technology was developed by Charles Wiseman, initially out of MD Anderson, and then went over to St. Vincent's Hospital in Los Angeles, and he developed the SVBR1-GM cell line. This is a cell line derived from the patient with breast cancer. And he would irradiate it and used it to treat patients to stimulate an immune response against the breast cancer for people who already had breast cancer. And he actually had some very interesting results initially, and then he decided to up the ante by putting the gene for a molecule called GM-CSF into the cell so it would produce this and basically work as an immune cell itself, stimulating the immune response even more. And Chuck did some very good work in a few patients a number of years ago. Unfortunately, as a research doc, his funding got cut off when St. Vincent stopped funding research, and it took a little time for him to connect with Saeed Babai, who's our chairman of the board, and engineer a reverse takeover into a Canadian mining company to create what is now BriaCell Therapeutics. So Chuck had some interesting data, and I'm happy to talk about that some more if you'd like.
Yes, I'd love to hear it.
So Chuck treated 4 patients with the SVBR1-GM cell line, and all 4 of them did better than you would expect in terms of survival. But one patient in particular had a really remarkable response where she had metastatic breast cancer. She had been treated with chemotherapy and initially responded but then relapsed. And so she came to Chuck and he treated her with this targeted immunotherapy and was able to induce nearly a complete remission in this patient, including her breast cancer in the breast, also in the lungs, in soft tissues, really a remarkable response. And then the study ended temporarily for her. They were only allowed to use 5 months of treatment at the time. But so she relapsed, and then he got permission from FDA to treat her again, and she responded yet again. Very interestingly, Marcus Latcher, a brilliant scientist, joined our company about 2 years ago and figured out why this patient was such a remarkable responder. And that has to do with a very widely available technology called HLA typing or tissue typing. Marcus figured out that this patient actually matched the SVBR1GM cell line at 2 different HLA types. And this is the same thing that's used for tissue transplants when you match somebody for their tissue transplant, for a kidney transplant, for example. So she matched at 2 different HLA types with the SVBR1GM cell line, and that told us that this may be a way to identify patients who are the most likely to respond. And it also taught us how in the future we can modify this cell line to match more and more patients so we can treat a wide number of patients with breast cancer.
Dr. Williams, you mentioned something very interesting in what you just said there with regard to the efficacy of the drug itself. Does this mean that the patient has to have ongoing treatment throughout their entire existence, or is this something you can move towards some form of reversal of the existing cancer? Because it seems like you use it on the patient, you got remission, then all of a sudden she doesn't use it again, and she gets remission again. What does that mean from a research and efficacy standpoint?
Yeah, no, I think it's a really good point. Now, you have to keep in mind that one of the big trends in immunotherapy today is to use combinations. So this was done basically with BriaVax in combination with a couple of other drugs that Chuck had figured out over the years work really well together with these targeted immunotherapies. And so she got this response, but then she relapsed. Now, today, we have more treatments available, things that are called checkpoint inhibitors. People may have heard of Keytruda or Opdivo or Yervoy. And these things have been out there on the market in a number of different tumor types. But they don't work in all cancers. They work in cancers that people say are hot cancers because they have a lot of cancer antigens, a lot of mutations that the immune system can recognize and then can reject the cancer. But there are colder cancers, and breast cancer is one of these, where there aren't that many mutations, that many things that the immune system can recognize. So what we can do with this targeted immunotherapy is also turn those cold cancers hot. So now the immune system's already recognizing it. And we hypothesize, we haven't shown this yet, that if we can then come in with some of these other immunotherapies, we really may be able to induce a sustained remission. But we're looking into that, even as we speak.
We've got less than a minute left. I know you've got a pipeline developed here, not just one drug, but we really don't have time to get into the entire pipeline on this conversation. We can have you back and talk about that. But before we go, there are investors out there looking to take a better look at your stock, want to understand the business side of your business. How can we reach out to our audience? How can you reach out to our audience and allow them to get in contact with you to really get into how you're saving humanity with your drugs?
Sure. So, of course, the easiest way is through our website, BriaCell Therapeutics. If you just put into the web browser BriaCell, that'll come up to our website. And you can contact us there if there's investment opportunities. Obviously, we're a small company. We're always raising money. But people can, of course, just invest on the open market as well on either the Toronto Exchange or the OTCQB.
Got it. BCTXF, right?
You got it. Or BCT on Toronto.
Got it. All right. Dr. Bill Williams, thank you so much for being a guest on our show.
Thank you, Michael.
You're welcome. Dr. Bill Williams, President, CEO of BriaCell Therapeutics Corporation, BCT on the Vancouver Exchange, and BCTXF on the U.S. OTC markets. Segment brought to you by Tycon Partners. We're going to go to a break now. You've been listening to CEO Money with Michael Yorba and Ed Baxter. When we come back, we're going to bring Ed Baxter in for the Private Equity Spotlight. We'll be right back.